my name is Lucas. I would like to present the HRF-Seq method, which is a method for assaying RNA backbone accessibility with hydroxyl radical detected with next-generation sequencing. Hydroxyl radical footprinting is a well-established method for RNA tertiary structure probing. It was used for studying RNA dynamics or for 3D structure modeling. Traditionally, the signal was detected with slab gel or capillary electrophoresis, which results in a low throughput. You can study only one molecule at a time and up to 400 nucleotides length. On the contrary, the presented here HRF-Seq method largely increases the throughput, allowing for probing multiple and long molecules simultaneously. The HRF-Seq experiment starts with RNA probing with hydroxyl radicals and keeping the untreated control. Both samples are subsequently reverse transcribed with the random primers harboring the Illumina adapter overhang. To the synthesized cDNA terminates on the probed site, and to those 3' termini, the adapter is ligated. The constructs are subsequently PCR amplified, pooled, and sequenced on Illumina HRF-Seq machine with the paired end protocol. During the experiment, in a PCR step, the primer introduces the sample-specific barcodes, allowing samples to be pooled. Additionally, in the adapter ligation step, the 7-nucleotide random barcode is introduced, which is useful for distinguishing the PCR duplicates from a real biological copies. Information from random barcode sequencing is used for estimated unique counts calculation. Estimated unique counts, or EUCs of a given fragment, are calculated based on number of unique barcodes observed for that fragment, but correcting for the possibility of independent barcodes having the same sequence. This kind of calculation was proposed before, but required equal probabilities of ligating different barcodes, which is not the case in our experiment. Therefore, we generalized the method, allowing for unequal barcode frequencies. EUC calculations are consistent with counting unique barcodes for lowly expressed fragments, but make a big difference for abundant ones. Use of EUCs helped to alleviate PCR and other biases, as indicated by preferential punishment of short fragments known to be overrepresented after PCR amplification. Additionally, the EUCs increased the overall quality of our dataset. We have compared the results of hydroxyl radical footprinting of RNA-sp specificity domain as detected with either slab gel electrophoresis or with HRF-Seq protocol. The quantification of the gel signal and EUCs show high level of correlation. Our next step was to measure the RNA backbone accessibility of E. coli 16S ribosomal RNA. After sequencing library preparation, sequencing, mapping, and EUC calculation, we obtained the information about priming site and termination site of each fragment. This information was used to calculate the termination count and coverage at each ribosomal position. The ratio between termination and coverage is called termination coverage ratio, TCR. TCR of the control sample is subtracted from TCR of the treated sample and yields delta TCR, which correlates well with RNA backbone accessibility as measured from the known crystal structure. Delta TCR signal displayed on the 16S rRNA structure underscores the high level of similarity between delta TCR and the known backbone accessibility. Moreover, the protein and large subunit footprints are clearly recapitulated. Helices displayed on ribosomal surface give clear signal indicating which site is protected. What's more, we see a global correlation of the signal coming from nucleotides separated by multiples of helical turn. Presented data validates HRF-Seq on known molecules. As a next step, we aim to probe the RNA accessibility in vivo. We have irradiated pieces of mouse liver with synchrotron-generated X-rays known to form in cytoplasm the hydroxyl radicals, which are able to probe the RNA backbone accessibility. Quality of our RNA decreases with increasing irradiation time over range from 0 to 1000 milliseconds, indicating that the irradiation worked as intended. Currently, we are preparing the sequencing libraries and will analyze in order to find a novel RNA tertiary structure which exists in vivo. We have shown that HRF-Seq readout agrees well both with the traditional method of hydroxyl radical signal detection as well as with the RNA backbone accessibility. To achieve that, we modified our experimental workflow, introduced the EUC concept, and proposed a normalization scheme. I'd be glad to answer your questions and discuss the method and its possible extensions or applications during this meeting or afterwards via email. Thank you for listening.